Hi, hello, good morning friends. Now we'll be start with one more class. In a previous class we have looked over the digestion part of the lipids, how the digestion process will be taken plus with the lipolytic enzyme. In today's class we will be start with the absorption of lipids, how the process of absorption of lipid will be taken place in a small intestine part. Okay, it's here. If you are looking for the nature of lipids, lipids are usually characterized by insoluble in water and soluble in organic solvent. But our GI tract is having a which nature? Aqueous in nature. It is rich with the water content or aqueous in nature. But lipids are insoluble in nature, insoluble in water. Okay, this opposite behavior of the lipids will be leads to some problem for the absorption mechanism. Even though this opposite behavior, our body system will be having some adjustment or compensatory mechanism, make that water insoluble lipid into soluble in nature. How this solubility will be increases, that will be see with the help of these steps. See here, first step, enzymatic digestion. Whatever the classes we have looked in a previous class, enzymatic action of the leakage over the lipids, or which are the enzymes will be act over the lipids means lipase. Primary is lipase and secondary phospholipase lipase A, phospholipase lipase B, and along with that cholesterol esterase. Those are the major enzymes which are active over the lipid parts. Okay, after the action of these lipolytic enzymes, finally we will be get a simple form of lipids, for example free fatty acids, monoacyl glycerols and diacyl glycerols, even though phosphoric acid and phospholipids. These are free form of lipids. And after that, one more mechanism will be taken place, that is emulsification mechanism. Here, larger part of the fat will be going to converted into smaller droplet, and this smaller droplet will be helping for the easily adjustable with the aqueous medium of GI mucosal layer or GI tract. And one more thing, what is the definition of this emulsification means? Emulsification is a process of dispersion of larger lipid molecule into smaller droplets. It is a dispersion of larger lipids into smaller lipid droplets. That will be called it as a dispersion process. And this dispersion process or emulsification process, how it will be carried out means by decreasing the surface tension of lipid molecule. By decreasing the surface tension of lipid molecule, along with this, it will be increases the surface area. Increases the surface area of lipid droplet. By looking this picture, this is a big molecule of fat. After the action of bile salt, it is going to become dispersed emulsified droplet, smaller droplet, and by this it will be reduced the surface tension. Surface tension will be reduced and smaller droplets will be occurred. And if you are looking for the area of the lipid droplets, it is usually increases. Area for this droplet, area for this droplet. Here so many droplets will be increases, and area of droplet is also going to become increases. By increasing area of surface area of droplets, easily acted by lipase enzymes, that a smaller droplet can be easily react with the lipolytic enzyme lipase and convert this smaller droplet into monoacyl glycerol or free fatty acids. Okay, this is our definition or simple process of emulsification. And one more thing, this emulsification will be taken place with the 3 steps. First one is the action of bile salt, and second is the surfactant action, third one is the mechanical mixing with the help of peristaltic movement of GI tract. Okay, first we will be start with the bile salt. For the digestion of the lipid, vital function is carried out by the bile salt only. This bile salt is secreted from bile juice along with the it is also synthesized in the liver. Liver along with the bile juice, or it will be secreted in a duodenal part, and this bile salt will be act over the larger molecule of fat, and it is also having the natures of 2 portions, internally hydrophobic portion, externally hydrophilic portion. That's why it will be adjusted with the both aqueous medium and lipid layers. Okay, make adjustment between the lipid layers and aqueous medium. And this bile salt act over the larger part of fat and make them to become an emulsified smaller droplets. And then further process will be taken place with the enzymatic action, and finally we will get a monoacyl glycerol or free fatty acid. This is how the bile salt will be act over larger molecule of fat. And second one, surfactant actions. What do you mean by surfactant action? Means it is a rearrangement of the fat molecules. Based on the requirement, rearrangement of the fat molecules will be taken place. Here, whatever the lipid portion is there, that is being with the central part, that will be called it as a non-polar part. Lipid is centrally enriched, and this is hydrophobic in nature. Okay, it is being center of the part, and that will be called it as a non-polar, and superficially covered by polar nature, and it is usually having the nature of hydrophilic in nature. It will be accept the water content and make the bond between the water and this lipid. Internally rich with the triglycerides, externally rich with the free fatty acids and phosphoric acid, some apoproteins, that usually will be called it as a mixed missile. Okay, this is how the surfactant action of lipid will rearrangement of lipids will be make the balance or adjustment between the lipids reach with the triglycerides and adjustment with the aqueous medium of intestinal lumen. By this only it will be become homogeneity. There is no dispersion. There is no rippling nature. It is a acceptance nature will be developed by the action of surfactant action of lipids. And the last one, third one, mechanical mixing. Here mechanical means vigorous shaking between the intestinal muscles, smooth muscles, with the help of peristaltic movement like to and fro motions. By this vigorous motion, whatever the fluid or food content being in the small intestine going to become vigorously mixed out, and molecules, larger molecules of fat will be become smaller things. With the help of these 3 mechanisms, total emulsification of fat will be taken place. After the emulsification process, then only process of absorption will be taken place. The process of absorption will be looking for this lipid means it will be follow 3 main vital things. First one is a simple diffusion, second one is a formation of mixed micelle, and last one is a formation of chylomicrons. Regarding absorption process of lipid is different from other carbohydrate and proteins. Here we should be kept in mind while absorption of fat, there is a must and should be occurrence of emulsification process and formation of mixed micelle, and lastly formation of chylomicrons. Okay, we will start with the simple diffusion. Whatever the simple form of lipids are there, which are easily dissolved in a water content, those will be follow the process of absorption of simple diffusion. Diffusion means there is a movement of molecule from higher concentration to lower concentration. Here higher concentration is being in a intestinal lumen and lower concentration being in a mucosal cell or blood capillaries. That's why movement of lipid molecules, simple lipid molecule from lumen to blood capillaries. Okay, this is about simple diffusion. And mixed micelle means these are the formation of mixed micelle. In the nature of this one, centrally filled with the triglyceride, superficially filled with the phospholipids, free fatty acids, apoproteins. And this mixed micelle will be helping for the transportation of short chain fatty acids to mucosal cells, from lumen of the intestine to mucosal cells. Whatever the short chain fatty acids are there, those will be get a help of this mixed micelle and diffuse into the mucosal cells. Okay, this is one of the vital thing. And next one, chylomicrons. For the larger lipid molecules or long chain fatty acids, we require a formation of these chylomicrons. These chylomicrons will be also having the same structure, centrally hydrophobic non-polar part, that is rich with the triglyceride and cholesterols, and the peripherally covered with the hydrophilic polar nature, that will be rich with the phospholipid, free fatty acids, simple cholesterol esters, and apoproteins. Okay, and whatever the long chain fatty acids are there, those will be attached with these chylomicrons, and those cannot be filtered into this venous capillary membrane. That's why those will be directly jump into the lymphatic circulations. This is the difference between mixed micelle and chylomicrons. Mixed micelle can be directly diffused into the venous capillary because this size is less as compared to chylomicrons. Chylomicrons will be having a long chain fatty acid and having a bigger size. That's why they cannot be enter the blood capillary. That's why those will be jump into the lymphatic circulation. From lymphatic circulation, it can be enter the chylomicrons to the systemic circulation. From systemic circulation, it will be reaches the liver, and liver will be carried out the upcoming metabolic process. Okay, this is how the process of absorption of lipid will be taken place, and these mixed micelle and simple diffusion mechanism will be whatever the fat will be diffusing venous capillary or intestinal capillary, easily taken out by the portal circulation to liver. But in chylomicron, it will be bypass the lymphatic circulation, then systemic circulation, then liver. Okay, by this, all whatever the complex form of food is there, that first has to be converted into simple form of lipids. After the simple formation, there is a three process of absorption: simple diffusion, mixed cell formation, mixed micelle formation, last one chylomicron formation. Through this process, liver will be take the whatever the digested fat is there and take into the site and make the deposition. Whenever the requirement, that will be made further variations or modification in the fat molecules that will become as a part of metabolism. By this we will be complete today class. In upcoming class, we will be look over the metabolic process of lipids. Okay, thank you so much.